Hello. Okay. Is it audible now?
Yes, sir.
Hello.
Yes, sir. Audible.
Okay. Okay then.
Good afternoon, sir.
Very good afternoon. Good afternoon. Good
Good
afternoon, everyone.
afternoon, sir.
Okay. In the last class we have studied. Again it asked, 6, 9, 6, 9, right? Task code is 6, 9. Eight, 4, please. Okay, just a second. 6, 9, 8, 4, 3, 7. Okay. Is it okay now? Yes. Yeah. Today we are going to discuss about the what topic? Do you know? Have an idea? Which topic you are going to discuss? Actually, in the last class, we discussed about the photosynthesis, and then Sunday, I could not able to attend. That class taken by Kumar Sharma. Sunday I taken respiration.
Inspiration.
Yeah. Today, we are going to discuss about the fourth unit, that is,
Plant growth and development.
yes, plant growth and
Development.
development. Okay. Is it visible to you?
Yes, sir.
Screen is visible?
Yes, sir.
Okay. Okay, then. Today, we are going to discuss about the what? What we are going to discuss?
Plant growth and development, sir.
Yes, in the plant growth and development, we are going to study the 4 chapters are there. In that 4 chapter we will discuss. Okay?
Okay.
Yeah, a 4 chapter, if possible, 2 chapters, and one more class also there now for us. In that class, we will discuss the remaining topics, and also in the photosynthesis, some topics are remain. Photo respiration, that is there now.
Yes.
That also, the remaining topic we will discuss in the next. Okay?
Okay, sir.
Today, as much as possible, we complete this. If otherwise not complete, we will discuss in the next. Okay?
Okay, sir.
Then, you know what the growth, what is the development? What is the growth? What is the development? Development means, growth means. What is the growth?
The growth is increase the size, sir.
Yes. Yeah. And development means? Are you able to see the screen?
Yes, sir.
Okay. Yeah. Development means is the overall metabolic improvement, metabolic action. Growth means only elongation, cell
Yes.
division or elongation. Okay. But whereas the development is overall development, that means physiological development. Physiological development, that means all the physiological reactions. That means development of photosynthetic reaction, respiration reaction, enzymes production, stress related, and nitrogen, phosphorus, sulfur, remaining all the metabolic reactions. That all reaction overall growth is nothing but development. Okay, that is the development. Okay, growth and development. Yeah, there is a need. You know, the plant growth is eternal. You know, plant growth is eternal. Eternal, or only sometime it will be. You know, our animals, all the animals, my camera is not visible. Okay, animals will grow up to certain stage only. But whereas plants, plants will grow
They
eternal.
grow lifelong.
Yes, till the death. Lifelong, eternal growth. It shows the
Throughout the life.
eternal growth throughout the life. Okay, that is the difference between the plants and animals. Plant cells, it shows the eternal growth, whereas the animal cells, it stop the growth after adult stage. Up to 21 age only, we having the growth. Okay, that is the difference between plant and animal growth. Okay, and then here in this chapter, we are going to discuss nitrogen and sulfur metabolism. First chapter is nitrogen and sulfur metabolism. How the nitrogen is doing the function, what is the importance of the nitrogen, how it is working, how it assimilating, what is the main function, what is the main source, how the biologically nitrogen fixation is taking place, how the biologically sulfur fixation is taking place, from where it is observing, and where it is participating in the metabolic reaction, where the nitrogen participating in the metabolic reaction, where the sulfur participating in the metabolic reaction. That all we will discuss. Okay. Okay. Then the first one is nitrogen and sulfur metabolism. Okay. Nitrogen and sulfur metabolism. Okay. What is the nitrogen? Where from, the in the atmosphere, how much amount of the nitrogen is there? 78% of the nitrogen is there. Yes or not?
Yes.
78% of the nitrogen. But there is no usage of nitrogen. No plants and animals can utilize that atmospheric nitrogen directly. Then what the usage of it? The nitrogen, atmospheric nitrogen is N2. It having the triple bond, strongest bond, strong bond. N2. Two nitrogens which are bonded with 3 bonds. That's why it is a strong bond. It could not be split by the plants and animals directly. So we can't utilize this nitrogen, atmospheric nitrogen. N2 is the atmospheric nitrogen that we can't use. Okay, but some of the microorganisms, bacteria and fungi, some of the algae, blue-green algae, bacteria, fungi, these three only. Hello? Is it audible? Okay.
Yes,
Bacteria,
fungi,
fungi,
blue-green,
and blue-green algae, cyanobacteria, that also cyanobacteria. Okay. These three only can use the atmospheric nitrogen. This atmospheric nitrogen gets converted into NO2 and NO3, nitrate, nitrite, and also ammonia. Okay. These three forms. It changed into these three forms with special effects of enzymes which are there in the microorganism, which are there in the bacteria, cyanobacteria. Okay. Because of that enzymatic reaction only, we could able to get this nitrogen. The nitrogen is the main metabolic inorganic molecule. Nitrogen is the main source in the proteins, amino acids, protein structure. Amino acid structure? Why is the resound? Amino acid structure?
Yes, sir. It is it contain ammonic group and carboxylic.
One end ammonia group, amino group, Other end is carboxyl group, COOH and NH2. One end NH2 group, other end COOH, carboxyl and amino, and in between CH. Okay. CH, NH2, COOH is the basic structure of the amino acid. These all the amino acids will combine together by which bond it is? Peptide bond. You know the peptide bond, polypeptide. COH in between the two. In between the two. COOH. Yes or not? COOH. One amino acid COOH, OH, and amino group 1H, H2O. It's released as a H2O. And what it remains? NHCO. 1H gone. That's why NH. CO, that means C double one O. Okay. This is the peptide bond. CONH. This is we call it as peptide bond. Okay, this is peptide bond. Like that, so many amino acids bind this peptide bond to form the protein. This is shown as no? Protein. So main building blocks are amino acids. Main building blocks are amino acids. That amino acid what's going on? This amino acid is the building block for what? Building blocks for protein. The nitrogen is the main building block for the amino acid protein. So that's why it needed the my camera is not opening.
So disturbance is there, sir. Shivalila ma'am, please mute.
From my side.
Sir, Shivalila ma'am.
Okay. Now is it okay?
Okay, sir. Okay, sir.
Yeah. Then we will go for the next. Okay. Where we are, what we are studying. First one is in the nitrogen. That is nitrogen is the main source of the amino acids and protein. Amino acids and protein. Okay. So there is a lot of importance, significance to the nitrogen. Though we could not able to observe, we could not able to get the nitrogen directly, but it is a very much needed. Whatever the protein is there in our body, that is made because of microorganisms. That microorganism converting atmospheric nitrogen into nitrate, NO3, NO2, and nitrite, NO3, and also as ammonia. You know the nitrogen cycle. You have studied already in the intermediate degree level. Nitrogen cycle you have studied. Nitrogen fixation, nitrification, ammonification, denitrification. 4 stages are there. How many stages are there? 4 stages are there in the fixation. That is the nitrogen fixation, also called as nitrogen cycle. Nitrogen cycle. Okay, where the eraser? Okay, here. Nitrogen cycle. That nitrogen cycle is the main. Here is the microorganism is the main source. Nitrobacter and nitrosomonas. Nitrobacter and nitrosomonas. These two are very, very important enzymes that is causing for the nitrogen assimilation. That means production. Nitrogen assimilation. Okay. Then the first chapter. Oh, is not showing. Okay. Here you can see what are those topics. Significance of nitrogen. We already discussed it. What is the significance? It is the main building block in the amino acids, proteins, lipids, except carbohydrates. In the remaining all the biomolecules. In the remaining biomolecules. Okay. Even enzymes, hormones, the vitamin, protein, amino acid, in all that. Okay. Nucleic acid, main nucleic acid. Okay. And you know, so many biomolecules, other biomolecules also. Okay. These all are main sources of the nitrogen in our body. Though it is not an actual atmospheric nitrogen, it is the base source for all the enzymes, amino acids, proteins, nucleic acid, and other metabolic products, secondary metabolic products. Okay. And the main nitrogen metabolism, ammonia production, is done by basic reaction, that is GS-GOGAD, glutamine synthase, glutamate, 2-oxy, glutamine transferase, GOGAD, glutamine, oxy, glutamate, amino transferase, GOGAD. GS means glutamine synthase. These two enzymes are there in the bacteria and in anaerobic, especially in anaerobic bacteria. You know the nitrogenase enzyme. You know the NIP genes. NIP genes and చదివే ఉంటారు మీరు. NIF, NIP. NIP genes. These NIP genes are there in the bacteria, and you know the root nodules.
Yes, sir.
These root nodules are present in the, where it present? In all the plants?
No, sir.
Leguminous plant.
Leguminous
Yes,
plant.
in the leguminaceae plant. All the pulses. That's why, you know, all the pulses having the richest protein. Why we are calling the pulses or proteinaceous food? Because of rhizobium bacteria. It observing the atmospheric nitrogen and it converting into ammonia. That ammonia is participating in the amino acid synthesis. That amino acids are forming in the protein formation by the peptide bond. So that protein formation is taking place in the pulses crop with the help of rhizobium bacteria. That's why symbiotic nitrogen fixation. That is called as symbiosis. Each other, helping each other. The plant that is giving the, what it is giving?
Food.
Oxygen and the photosynthetic pigment, photosynthetic products to survive the rhizobium bacteria. Rhizobium is providing ammonia. That ammonia is taken by the roots and it using in the formation of protein. Okay. That the symbiotic nitrogen fixation. Okay. And you know the what it is. Significance we already completed. Biogeochemical cycle. Next is biogeochemical cycle. Next is nitrogen fixation. Next chapter we will go for the biogeochemical cycle. You see here, what is there? Could you able to see this?
Yes, sir.
Yeah. Now enlarge it. You see, nitrogen in the as many as resources. You see, biological nitrogen fixation, industrial nitrogen fixation, electrical nitrogen fixation. You know, at the time of thunder, urumulu merupulu antama.
Yes.
At the time of thunders also, atmospheric nitrogen with the ozone, O3, atmospheric nitrogen plus O3, it can convert into nitrate. Thunder also useful to form the nitrate and nitrite, NO2 and NO3. You know, the plants absorb only nitrate, nitrite, or ammonia. These three forms only. Atmospheric nitrogen, they can't absorb. Whatever the form, these three forms, plants can absorb, and it will be utilized in the protein synthesis. Okay. And denitrification. You know the denitrification? Denitrification means again formation of the atmospheric nitrogen, either from the ammonia or from the nitrate. Again, it is going to atmospheric nitrogen, N2. Okay. These all again participating in the metabolic reaction. Whatever the ammonia, whatever the nitrates that are formed, that all uptake by the root, and this will be utilized in the protein synthesis. Okay. You can see here the nitrogen cycle. It is a nitrogen cycle. What are there? First is the nitrogen fixation. N2 to NO2. Okay. First one is the nitrogen cycle fixation. What are those four steps? Nitrogen fixation, ammonification, nitrification, and denitrification. Okay. Have you understood? What are those four steps?
Nitrification, ammonification,
Fixation, nitrification, acid.
denitrification,
Ammonic.
Yes. Nitrogen fixation. To draw na. Nitrogen fixation. This is black color in to change the color. Where the color? Okay, little bit. You see from here, nitrogen fixation, ammonification, whatever the ammonia that assimilate in the biological product, nitrification, and again denitrification. Okay, and again denitrification. Okay, these are the four steps. You know, we are these are all the steps, or otherwise we will discuss. So, and it is not moving. Okay, here you can see clearly. Okay, here you can see very clear. Okay, what are those steps? The first, nitrogen in the atmosphere, that is taking place in the nitrogen fixation, that is forming the ammonia, nitrogen to ammonia, and then again what happened? Nitrification, that converting into NO2 or NO3, and again NO3 is going again to atmospheric nitrogen. Okay, these are the four steps which are there in the nitrogen cycle. You know, we all studied the biogeochemical cycle: carbon cycle, hydrogen cycle, nitrogen cycle, sulfur cycle, phosphorus cycle. You know, sulfur and phosphorus cycle are solid state cycle. Carbon, hydrogen, nitrogen, oxygen is the gaseous cycle. In the form of gases. Okay. Phosphorus and sulfur are rock. That means solid state. Okay. You see, whatever the nitrogen assimilation is taking place in this four steps, four different processes, nitrogen is accumulating, that means assimilating in the biological function. You see, these are how they are. Okay. You all know the meaning of it now. Nitrification, ammonification, you all know now. Again, I have to explain now. Nitrogen fixation means whatever the atmospheric nitrogen that get converted into ammonia. That is the nitrogen fixation. Okay. Either it is biological or by physical. Okay. Biological or physical. You know the biological again types are there. Asymbiotic and symbiotic. Pre-nitrogen fixation. Pre-nitrogen fixation occur in so many competitive exams adigaru. Pre-nitrogen fixation. Pre-nitrogen fixation means there is no symbiotism, there is no assistance with others. Only the bacteria which directly synthesize the ammonia from the nitrogen. You know, what are those? Azotobacter, azotobacter and acetobacillum. Okay, that are free-living bacteria. Whereas rhizobium, branchia are symbiotic. By symbiont with the plant, it produces the nitrogen. It do the nitrogen fixation. Okay, that is biological nitrogen fixation. And the pre-nitrogen fixation is by azotobacter, azospirillum. This directly can absorb the nitrogen and can convert into ammonia. Okay, these are the types. Okay, N2 plus O2, NO, and again mixing with the oxygen, NO2. Okay, and again if it is mixing with the water, rainwater, that is converting into nitrous oxide. That means nitrous oxide. That means HNO2 or nitric oxide, HNO3. Okay, and then the next one is ammonification. That means whatever the organic nitrogen, that means in the dead body or in the dead plant, by decomposing, that whatever the ammonia is there, whatever the protein building blocks ammonia is there, that ammonia through microbial decomposition. My camera is not visible. Microbial decomposition. Okay, by microbial decomposition, that decompose the mamsam protein and they separate the what it separate? Ammonia from the organic compound. That means from the protein, it separates the ammonia. That means ammonification. From the protein, mamsam, muscle, okay, skin muscle, whatever the bioproduct, organic nitrogen product, from the organic nitrogen, by decomposing, it converting into ammonia. That is ammonification. Okay. And then next is the nitrification. Nitrification means, can anybody? Nitrification means? Nitrification means?
Ammonia converted into nitrate form.
Yes. Ammonia, whatever the ammonia formed in the ammonification, that convert into nitrate. And what? Okay. That is called as nitrification. That is called as nitrification. Okay.
The formation of nitrate and nitrate is called nitrification.
First it form the nitrite and they get adding another oxygen molecule, it convert into nitrite, O2, O3, 2 oxygen, and again by adding the other oxygen, 3, NO3. Okay. You know the nitrite is formed by nitrosomonas. What it is? Nitrosomonas bacteria. Nitrosomonas bacteria. And nitrite formed by nitrobacter. Nitrobacter. Nitrobacter convert nitrite into nitrate. Nitrosomonas convert into nitrite. Ammonia into nitrite. Okay. Is it clear now? This all the process is called as nitrification. Okay. Okay. And from the amino acids, proteins, muscles, dead bodies, decomposing material, splitting of the decomposing material, formation of the ammonia is called as
Ammonification.
okay. Okay. This is first one, nitrogen fixation, atmospheric nitrogen to ammonia. Second one, from the dead bodies decomposing material, organic nitrogen to ammonia. And third one is nitrification from ammonia to nitrate and nitrite. Okay. And the fourth one is denitrification. That means again going back as a atmospheric nitrogen. This step. NO3 is converting into atmospheric nitrogen. N2. Denitrification. You know, thiobacillus denitrificans. Thiobacillus denitrificans bacteria. This convert the nitrate into nitrogen molecule. Atmospheric nitrogen. Okay. That means it is splitting the oxygen molecule. Splitting the oxygen molecule. Separating the oxygen. NO3 to NO2. NO2 NO. NO2 N2. Okay. NO3 to NO2. NO2 to NO, nitrous oxide. Okay. Nitric oxide, nitrous oxide, okay. Then nitrate. Nitrogen, not nitrate. Okay. They are separating the oxygen molecule from the nitrate. Okay. That is denitrification. Again forming the atmospheric nitrogen. The cycle. This is the nitrogen fixation cycle. Very important. Ask the question. Okay. And also nitrogen fixation. Earlier I have shown you how many ways the nitrogen fixation is taking place. Biological, industrial, atmospheric, atmospheric, and by natural. Thunders. Urumulu merupulu vachinappudu. By the 4 ways, the nitrogen fixation will take place. Okay. I have shown you now here earlier. You see, in these 4 ways, nitrogen assimilation, nitrogen fixation will take place. Okay. Okay. And then here also you can see. Very good. Okay. In the 4 ways, the nitrogen assimilation is taking place in the atmosphere. Okay. And then you can see what is the importance of the nitrogen. I already told you. Essential as the essential nutrient, amino acids, proteins. In the photosynthesis, also it's needed. Why? In the chloroplast, Nitrogen is there now. We have studied 4 pyrrole links. 4 pyrrole links it having the nitrogen. That 4 pyrrole links mix combined with the which one? Which one? Magnesium. If it is mixed, at the middle is the ferrous, that is hemoglobin. Okay, hemoglobin in our blood. If it is the magnesium, that is chloroplast in the plant. Whereas the hemoglobin, it absorbs the oxygen. Here in the chloroplast, it releases the oxygen. Okay, that is the quite opposite. Okay, same pyrrole ring, but at the middle is the magnesium, that is chloroplast. You know, chlorophyll A, chlorophyll B, we studied. Chlorophyll A, chlorophyll. 350 by H72, O5, N4, MC. We have studied in the photosynthesis topic. Yes or not? Forgot, huh? Okay. Then nitrogen fixation organism. Some examples. Rhizobia, legume, symbiotic. You see what are there in the soya beans usually? Branchia, where in the branchia? Where is the rhizobia? In which organism? What type of the bacteroid? You know, rhizobiums we call as bacteroids. That means bacterial symbiotic organisms, bacteroid. Okay, here you can see the various 4 ways of the nitrogen fixation. You see, pre-living nitrogen fixation. Already told you what are those? Azotobacter and azospirillum. Okay, anaerobic bacteria. What are anaerobic bacteria? Pre-living nitrogen fixation. What are these? Clostridium and Dromibia. You know the Clostridium. Usually, whenever we get infected with the soil injuries, that cause for the tetany. Dhanurvatam antama? That is caused by Clostridium. But it is usually a lot of Clostridiums are there in the soil that converting the atmospheric nitrogen into ammonia. Okay. అవి ఉన్నాయి కదా అని మనం గాయాలు తగిలించుకుంటే ఆ గాయాల నుంచి క్లాస్ట్రీడియం లోపల పోతుంది, మనకు టిటానస్ వస్తుంది. ఓకేనా? And then you can see facultative. What are those? Klebsiella, Azotobacter. You know, obligate facultative. We have studied in the first semester. Obligate parasite, facultative parasite. Okay. And aerobic bacteria, Azotobacter. And then what is that? Photosynthetic bacteria, Rhodospirillum, Chlamydium, Clostridium. These are photosynthetic bacteria. These are pre-living organism that fix the nitrogen. Cyanobacteria, Anabina, Anastar, the symbiotic. Okay. And like that, so many are there that you all know. We will see the later. Okay. You already studied that all. We having only half an hour. Oh, we have to complete. Okay. You know the nodules formation. How the root nodules of formation is taking place. Root nodules. You know, whenever the rhizobium bacteria, it enter toward the root, then it penetrates into the root tissues through the stomata, not the stomata, where the epidermal cell tissues. Here, it releases the lutein enzyme. That lutein, because of that lutein enzyme, it splits the epidermal cells, and it rhizobium will enter inside. Will enter inside. Whenever it enter inside, it form the bulges. That means it bulges, swollen, bulges. Then it form the layer. You see, whenever it goes inside, it is enter at the middle, and it form the seed. Okay, that seed we call it as Hello, prebacterial membrane or peribacterial membrane, which cover the bacteria. That's why peribacterial membrane. Okay, that peribacterial membrane will protect the rhizobium from the root exudate, whatever the root it exudate to kill the foreign bodies. But it will be protected with this peribacterial membrane. And then whatever the inside rhizobium, that we call as bacteroid or rhizobioid. These bacteroids or rhizobioid will be prevented from the oxygen. You know, this nitrogenase enzyme will be active in anaerobic condition, absence of oxygen. In absence of oxygen only. If oxygen is present, we can't do the nitrogen fixation. So it created the special chamber and that having the anaerobic condition, so that it is very easy to do the nitrogen fixation by the rhizobium, so that it forms the root nodules. Okay, that root nodules, you see the root nodule structure, how it is. You see, could you able to see it clearly? See, root nodules. These root nodules having the peribacterial layer, so that it forms the anaerobic condition inside, and it having the nitrogenase enzyme, NIP genes inside. Okay, in the nodule, root nodule. Where budipelu antama? Root nodule. Okay, in this nodule, nitrogenase enzyme will be activated. Then it starts the nitrogen fixation in anaerobic condition. Okay, that function, how it is, you can see here. These are the 3 steps. How many? 3 steps are there. For that, it needed molybdenum ferrous complex. That means MOFE. What it is? MOFE, molybdenum. You know, in the nitrogen fixation, molybdenum is very much essential. If there is no molybdenum, nitrogen fixation will not take place. As like there is no magnesium, there is no releasing of the oxygen. As like there is no iron in the blood, there is no absorption of the oxygen. Okay, you know, essential elements are non-peristout essential elements, 16 essential elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, magnesium, sulfur. These are the macro elements. And micro elements also there, na? In the water mineral nutrition you have studied, the first unit. First unit already physically completed, na? There you have studied mineral nutrition, essential elements. Okay. And ferrous, manganese, zinc, cobalt, copper, that all are micro elements. Okay. Macro 9, micro 7, total 16. Okay. Here is the molybdenum. Molybdenum is present in the nitrogenase enzyme. Nitrogenase enzyme is a complex enzyme. biological nitrogen fixation, biochemistry. Biological nitrogen fixation is take place by nitrogenase enzyme. What it is? Nitrogenase enzyme. This nitrogenase enzyme is not a simple enzyme. It is a complex enzyme. It having the coenzymes, okay, cofactors. What are those cofactors? Molybdenum Perrous protein factor and Perrous protein complex. Two complexes. Complex. This is the complex 1. This is the complex 2. Two complexes are there. With these two complexes, atmospheric nitrogen is absorbed by the nitrogenase enzyme in the root nodules. Whatever the hydrogen molecule, protons, that will be added here to the atmospheric nitrogen. Okay. That atmospheric nitrogen adding the hydrogen molecule, you know, here it needs total how many? 16 ATPs are needed for this reaction. How many? 16 ATP. How many ATPs are needed? 16 ATPs, 8 electrons and 8 protons. For one atmospheric nitrogen converting into ammonia for one N2 to ammonia, one N2, one ammonia. This is the nitrogenase enzyme complex which is participating, converting the atmospheric nitrogen into ammonia. It's a complex enzyme. It having the two complexes. One is the molybdenum Perrous, another one is the Perrous sulfur complex. Okay. Then here it is adding the first one, it is adding the proton, that means H plus ion, which are there from where it is observing, you know, in the photosynthesis. Photosynthesis, light reaction, dark reaction, NADPH2, FAD, are formed or not?
Yes, sir.
Kelvin cycle. Yeah, started. There, whatever the FADH2 and NADH2, reduction causing enzymes, reductase enzymes. That reductase enzymes, NAD reductase or FAD reductase. That means succinate reductase or pyruvate reductase. Pyruvate reductase is NADH2, succinate reductase is the FADH2. These two participating and is releasing the proton and electron. These protons and electrons observing by nitrogen and it converting into ammonia with the help of 16 ATPs. It needed energy. That energy is observing by the ATP. That ATP is also formed in the photosynthesis. So for the nitrogen fixation, chloroplast is the basic source. Initiation of the nitrogen fixation, nitrogen assimilation will take place or it initiates in the chloroplast. Whatever the secondary products, they are releasing alpha ketoglutarate amlamu acid, pyruvic acid, glutamic acid, that are using here to synthesize the ammonia. Okay. Second one, you know, we already studied. Okay. First one is MOFE complex. Second one is FES complex. MOFE complex, it observes the atmospheric nitrogen and it get with participating in the molybdenum ions. And whatever the electrons release, that electrons sending it to here, and protons also sending it here by splitting the NADP, whatever it is, NADP H plus, and then that is observing here, and here it is converting into ammonia. Here pyridoxine molybdenum and pyridoxine. These two carrier proteins. Pyridoxine is a carrier protein. Plastocyanin and pyridoxine. These two are the carrier proteins that you have studied in the ETS, electron transport system. Respiration law. In the respiration, ETS, electron transport system, respiration law. Those two, that mean in the photosynthesis, respiration, nitrogen metabolism, these three are combined together. One reaction affecting other reaction. Photosynthesis affecting respiration, photosynthesis and respiration affecting the nitrogenation. Okay. This reaction you have to write. That is also called as GS-Gogat reaction that we will explain detail in the next slide. Okay. Here you are known of two complex, two complexes. Okay. The two complexes causing the preparing the ammonia. Okay. Then where we are. Okay. Here is the all reaction. Association with the photosynthesis, respiration. Whatever the alpha-ketoglutarate acid, oxalosuccinic acid that are released in the respiration that are being used in the nitrogen solution. Whatever the photosynthetic products, pyruvic acid, oxaloacetic acid, that also being used in the nitrogen solution. That means photosynthesis, respiration, both are involving in the nitrogen assimilation. Nitrogen assimilation. Yeah, here you can see. What is the basic? Very, very important question. GS-Gogat. What it is? GS-Gogat pathway. Okay. GS means glutamine synthase. Gogat means glutamate synthase 2-oxy glutamide amino transferase. Okay. These two enzymes. First GS, it's converting glutamic acid. Whatever the glutamic acid is formed, that forms into glutamine by adding the ammonia. By adding the ammonia. Glutamate plus ammonia with the help of ATP.
ATP.
It is forming the glutamine. Amino acid. Here amino acid formation taken place. You know, amino acid formation also very, very important question. Amino acid formation will take place in the two ways. What it is? Reductive aminase and transaminase. Reductive aminase and transaminase. Reductive aminase means whatever the reduction, either from the NADPH or FADH2, H plus molecules, adding to the nitrogen and it forming the ammonia. Okay, first step it is nitrogen fixation. In the nitrogen fixation, atmospheric nitrogen converting into ammonia. That as like that, here first acid, two types. What are those? First one is reductive aminase by doing the reduction reaction. You know, oxidation, reduction. Oxidation reaction, reduction reaction is opposite. Okay. If oxidation number is increased, that is oxidation reaction. If it observe oxygen, that is oxidation reaction. Okay. Have you understood? If oxidation number is decreased, 3 plus 3 2 plus 2 plus 2 to 1, decrease, that is reduction reaction. Okay. Or if otherwise it observing the hydrogen, that is a reduction reaction. Okay. Oxygen, releasing the oxygen or observing the hydrogen. Okay. Observing the oxygen, releasing the hydrogen. Two quite opposite reaction. Okay. One is the reduction, other one is the oxidation. Okay. Oxidation reaction, Oxidative phosphorylation you have studied in the respiration. By releasing the protons, H plus ions, it forming the ATP. That means it's losing the H plus ion. That is oxidation.
Oxidation.
Okay. Understood?
Yes, sir.
Yeah. There in the respiration, oxidative phosphorylation. In the photosynthesis, photophosphorylation. In the respiration, oxidative phosphorylation.
Oxidate.
Okay. These two phosphorylation utilized in the amino acid formation. Okay. The first basic amino acid form is glutamic acid. That means glutamine. Okay. This glutamine is participate in now what is the next second stage reaction? Transamination. Amino group transforming from one to other. Here you see glutamine amino group added to
Alpha ketoglutarate.
yes, alpha ketoglutarate. Then again it forms the glutamate. Okay. That is the Gogat reaction. This is the GS reaction. This is the Gogat reaction. That means here two. Second place oxygen. Second place it is oxygenation is happening. That's why glutamate to oxo, glutamine, amino transferase. Okay. Second position of the glutamine, oxygenation is happening. So that and then amino group also transferring. Amino transferring. Amino group transferring from glutamine. That's why glutamine amino transferring. GOGAT. GOGAT. This is the GS GOGAT reaction. You see here clear. First one is the GS reaction. Second one is the GOGAT reaction. You see, glutamate adding the ammonia form the glutamate. Okay. This is the reductive amino acid. GOGAT reaction is transamination. You see here. Okay. Glutamine with the help of NAD. Okay. What it is? Alpha
ketoglutarate.
Okay.
Alpha keto.
Glutarate. Okay. That is the GOGAT reaction. Okay. These two reactions are very, very useful. With this reaction, not only useful for the ammonia formation, but also again oxidation and reduction reactions, and also maintain the pool of protons inside the chloroplast and mitochondria for the ETS, electron transport system, that will be utilized for the ATP formation. Okay, this link reaction, photosynthesis, this nitrogen assimilation, GS GOGAT, is linked with the photosynthesis and respiration and maintaining the balancing of the cell. Proton balance, H plus ion balancing inside the cell. Okay, there is the GS GOGAT. Here I already told you nitrogen, how many ways nitrogen is being utilized. Photosynthesis, respiration, growth, and remaining metabolic reaction, protein formation. So many reactions. Okay, these are we already completed. These are utilized in the we already studied. Okay, nitrogen completed. Then we go for the sulfur. Okay, sulfur. You see, sulfur also being utilized for so many metabolic reactions. You see, for the mineral absorption and assimilation of the energy, protons, and translocation of the water and fluids in the phloem, and even in the decomposition, even in the mineralization, and volatilization, okay, ionic formation, volatilization. Okay. In these so many ways, sulfur is being utilized. These are the 4 ways the sulfur assimilation is taking place. The 4 ways. What are those? You read out these 4. What are these 4 process? Read out.
Reduction
Ah.
pathway, amino
Yes.
acid synthesis,
Yeah.
antioxidant function, cellular
Yeah.
transport.
Yes. Basic major is the reduction pathway. Main in the reduction pathway, sulfur assimilate in the plant body, plant or animal. Okay. Main the reduction with the reduction reaction. That means NADPH2 to NAD, FADH2 to FAD. By releasing the hydrogen, that will be absorbed here, then converted to utilize useful sulfur. That means whatever the sulfur, it will be converted into H2SO3 and H2SO4. Sulfurous acid, sulfuric acid. You know, H2SO3 or H2SO4 form only will absorb by the plant. No sulfur directly absorbed by the plant. Okay. Sulfur is absorbed as a sulfurous acid or sulfuric acid. Sulfur by participating in the reduction reaction, adding the hydrogen molecule, so that sulfurous acid and sulfuric acids formation is taking place. Okay. And then assimilatory sulfate reductase. Whatever the sulfurs inside, that also will be utilized with the help of ATP ion. With the help of ATP ion, ATP dependent, especially in the green sulfur bacteria, purple sulfur bacteria. Okay. And then what happened here? Here it formed the APS. That means sulfur adding with the ATP, it forming the APS. ATP plus sulfur gives rise to APS. What it is? How to draw? Already you know. ATP. ATP, it takes time. Okay. ATP plus sulfur. It is forming the
ATS,
APS.
APS.
APS. This APS form easily absorbed by the
Plants.
organelle, cell organelle. Okay. Why? Because it energized sulfur. Energized. You know, ATP is energy. Basic energy source is ATP. It added sulfur added with the ATP. That's why APS form. Okay. Adenosine S2 phosphosulfate. Adenosine phosphosulfate. This adenosine phosphosulfate will easily absorb by any molecule of the plant. Okay. Once APS formed, easy to absorb by the plant molecule. Then it can convert into sulfur amino acid. You know which amino acid which are having the sulfur? You know 3 amino acids which having the sulfur.
Methionine. Methionine.
Yes, and then some more. Cysteine, cystine, methionine. Cysteine, cystine, methionine. Okay, these three amino acids form with the help of APS. With the help of APS. The 3 amino, you know, we need 20 essential amino acids. We need 20 amino acids. Okay, of that 3 are this one. Okay, methionine, glutathione, cysteine, cysteine. Okay. You know, whatever the sulfur bacteria, we call it thio. Thiobacter, which are using the sulfur. That bacteria we call as thiobacter. Sulfur means we all are pronouncing as a thio. Okay. Thio. That means methionine, glutathione, glutathione. Okay. You know, thiobacillus, sulfur having bacteria. Thio. Okay. Here sulfur, and then dissimilatory is sulfate reduction. Dissimilation. That means dissimilation means assimilation, dissimilation. First we have studied assimilation. This one is the dissimilation. Dissimilation means, you know, H2S, మురిగిన కోడిగుడ్ల వాసన వస్తుంటుంది. హైడ్రోజన్ సల్ఫైడ్. Yes or not? Lab లో మురిగిన కోడిగుడ్ల వాసన వస్తుంది. H2S.
Yes.
Sulfide. That can be converted into sulfate. Okay. With the help of APS. Especially ferrous sulfur protein. Ferrous sulfur protein. Earlier we have studied in the root nodules. Two complexes. FES complex and molybdenous ferrous complex. That FES complex will be useful in the dissimilation. That means inorganic sulfate to organic sulfur. Okay. By releasing the water molecule. Okay. And then oxidation reaction. By oxidation, I already told you. మురిగిన కోడిగుడ్ల వాసన వచ్చేదని మంచి గంధ వాసన వచ్చేటట్టుగా చేసేస్తుంది. That is I already told you. Thiobacillus, Bigieta, Bigieta, Thiobacillus. This convert the decomposed smell into good smell. Sulfur gandham. Gandham ఎంత మంచి స్మెల్ వస్తుంటుంది. H3S ఎంత బ్యాడ్ స్మెల్ వస్తుంటుంది. That's why the sulfur also very much needed. The Thiobacillus and Bigieta, these all are very much needed to clean the atmospheric environment from H2S to sulfur dioxide. Sulfur dioxide. Okay. Or sulfuric acid. H2SO4. Okay. These are so many significances also there that I will show you. ఒక చాప్టర్ కి ఏమన్నా అయిపోతుందిగా, ఇంకో నెక్స్ట్ చాప్టర్ చెప్పాలి. ఫైటో హార్మోన్స్ కూడా ఉంది. So many lot of key features are there for the sulfur. What are those features? The 4 features are there.
Synthesize the amino acid and proteins.
And
Plant stress response mechanism.
the stress
Chlorophyll synthesis.
even in the
Photosynthesis.
and contribution to the
Production of secondary
metabolite production.
defense.
4 ways the sulfur will be utilized. Very, very important. Okay. In the main key roles, in the nutritional role, physiological role, and environmental adaptation, and biochemical pathways. Biochemical pathways. That means sulfur having amino acid formation. Cysteine, cysteine, methionine. Okay. Here you can see nitrogen and sulfur, these two interconnected. Whenever these two are interconnected, the metabolic reaction is run in the highway. Okay. So many problems if sulfur lost or if nitrogen lost. Huh? Okay. Where we are. Okay, sulfur, the 4 ways and significance of the second sulfur also we have studied. Okay. And then next is plant growth hormone. You know the plant growth hormone? Plant growth hormone, growth regulators and growth inhibitors. Auxin, cytokinin, gibberellins are growth regulator. Abscisic acid and ethylene are growth inhibitor. Okay. Plant growth development. In the what is that? Plant growth hormone. Okay. The plant growth hormone, which are synthesized in the metabolic pathway. Plant metabolic pathways that are very much essential to the plant growth and development. Though these are very little amount, minimal amount, it acts much. You know, hormones always in the small amount, but it impact big. Hormones, it impact very big. Okay. You know, basically, we also call it as phytochrome, phyto hormones are plant growth regulator. Plant growth regulator. You know the how many? Can you ask me? It is not opening. Just stops here and then so. Where it is. Okay. Meanwhile, we will discuss by searching. Okay. The growth regulators and growth inhibitor. Growth inhibitor. Okay. Growth regulators are auxin, gibberellin, and
cytokine.
cytokine. Okay. And growth inhibitors are
ABA, ethylene. Abscisic amlam, ethylene.
Okay. Okay. How can I share this? Is it visible?
Yes, sir.
Okay. Is it visible now?
Yes, sir.
Plant growth hormones.
Yes, sir.
Okay. You can see auxins, gibberellins, and cytokinins, ethylene, and abscisic acid. Okay. These are very much useful in the growth and development, and also in the nutritional function, elongation, fruiting, flowering, and all the metabolic reactions. It impacts all the metabolic reactions. We know the difference between vitamin and hormone. Okay. Who is that? Who said the phytohormone? Who said? Timman. Who is that? Hormones of plant is called as phytohormone. Okay. Hormones, you know, the hormones means stimulate, which stimulate, that is called as hormone. You know, our hormones, adrenocorticotropic hormone, thyroid, stress hormones, FSH, LH, these all are there, na? Phytohormone. These are naturally produced in the low concentration, but it calls for the increase or decrease or modify the growth and development. These are the growth hormones, growth promote, and growth substances, growth regulators, growth inhibitors. What are those growth regulators? We already studied. Auxin, gibberellin, and cytokine. Okay. You know, the auxin, which is forming from the tryptophan. Tryptophan is the precursor for the auxin. Tryptophan, that is the one of the amino acid. Tryptophan is the one of the amino acid. From that amino acid, oxaloacetic acid. Oh, now it came up. My foot. Okay. Oxaloacetic acid form. That oxaloacetic acid, not oxaloacetic acid, indolacetic acid. Indolacetic acid, IAA. That is the first hormone identified. That is a very much useful hormone. Chalodny-Went theory is there. Auxin to establish the auxin action reaction. You know which is the natural auxin? Which is the natural auxin? So many competitive exams they ask the question. IAA. You know the weedicide, herbicides. తెలుసా herbicides? కలుపు మందు.
తెలుసు సార్.
కలుపు మందులు వెళ్తారు కదా. That are nothing but auxins. That are auxins. Okay. Auxins, especially synthetic auxins. That means artificial auxins. That we are preparing by adding the other chemical to the IAs. Okay. That is IBA, NAA, and 2,4-D. Okay, 2,4,5-T. IBA, NAA, 2,4-D, 2,4,5-T. These four are artificial auxins. Okay. IA is the natural auxin. This artificial auxin we use for the weedicide as a weedicide, as a weeds controller, herb controller. We use as a weedicide and herbicide to kill the weeds. Especially, it stops the photosynthesis. Then it automatically dries. There is no photosynthesis. There is no food formation. There is no oxygen. But IAA, indolacetic acid, we use as a growth hormone. Auxin, it means grow, increase. Auxin means grow, increase. To increase the stem size, to increase the branches, stem, and to elongate the stem, we use this auxin. Especially if we applied auxin, stem will be elongated. Much more branches will be formed. For the more crops, for the multiple branched plants, we use the auxin. Okay. Now for the root, you know, if we cut any plant, that cutted plant with a single node, where it is? If suppose some of the branch with the single node, we cut. The cut part, if we dip in the auxin, then it forms the roots within the 2 to 3 days. No need of the plantation, only rooted plants. With the single branch, we can produce multi-number of the new plants. By applying the auxin. If we apply the auxin at the end, the typical part, cut places, that part will become the clone. That means it having the root and it grow as a plant. That is the speciality, very, very important of the oxygen. Very, very important. Oxygens are very, very important. Okay. You know, Vent Avena curvature test. Vent, what he has done? Actually, oxygens are at the typical place. At the typical place. Vent, what he has done? He cut the typical part, and at that place, he used the sponge, so that whatever the oxygen that absorbed into the sponge, that sponge he kept in another plant, one side, not the full, one side. Other side it left. Okay. Then what happened? Where this part with having the oxygen, that side stem growth increase the stem growth. That means oxygens, where the oxygen that is causing for the growth, elongation, cell division. Oxygen it is causing for the more cell division, so that it's forming. You know, photoautotropism. మీరు ఎప్పుడైనా చూసింటారు, నీడలో పెరిగే మొక్కను చూడండి, అది ఎట్లా ఉంటుంటుంది? డెలికేట్ గా ఉంటుంది. ఎటైతే సన్లైట్ వస్తుందో అటువైపు టర్న్ అవుతుంటుంది. చూశారు ఎప్పుడైనా? నీడలో పెరిగే మొక్కని, ఒక మొక్కని మీరు పందిరి కింద ఎక్కడైనా వేస్తారు అనుకోండి, అది ఎటైతే సన్లైట్ ఉందో అటువైపు కర్వ్ అవుతుంటుంది. Photoautotropism. That means it attracting toward the light.
Sunlight.
That is the reason. Whenever where there is a photo light, that side oxygens are accumulated, so that it takes the more cell division. It grows toward the light. It grows toward the. That is nothing but vent theory. Charlotte Vent proposed this theory. That is a Charlotte vent theory. Okay.
వెళ్ళి పిలుచుకొని రారప్ప. అడిగావా నాన్న?
What happened? Time up. టైం అప్ప అయిపోయిందా? వాయిస్ వినిపిస్తుందా? రికార్డ్ లోనే ఉందా?
వినిపిస్తుంది సార్.
వినిపిస్తే లేదు, కట్ అయిపోయినాక కూడా నేను చెప్పుకుంటూ పోతే. మొన్న ఏమైందంటే కట్ అయిపోయింది, అయినా నేను చెప్పుకుంటూనే ఉన్నా. మీకు ఫోటో రెస్పిరేషన్ కూడా చెప్పేసినాం అన్న, కానీ మీకు రాలేదు. నేను ఒక్కనే చెప్పుకుంటున్నా. అట్లా అయిపోయింది మొన్న. నేను అయిపోయింది అనుకున్నా. ఓకే. దెన్ వాట్ ఆక్సిజన్స్ కదా. అయ్యో 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 చెప్పింది. Okay. Occurrence of the oxygens, where it already told you at the typical part. Structure of the oxygen, IAA, indole 3 acetic acid is the indole group. At the third place, it having the acetic acid complex. That's why indole 3 acetic acid. Okay, 1, 2, 3. Third carbon having the acetic acid. That's why indole 3 acetic acid. This is the primary structure of the IAA. Okay, and you know, these all are artificial oxygens. Indole butyric acid, indole propionic acid, naphthalene acetic acid, NAA, 2,4 dichlorophenyl acetic acid, 2,4,5 trichlorophenoxy acetic acid. These are artificial oxygen. These all we using it for the as a weedicide or herbicide. You see NAA structure, IPA, IBA structure, indole butyric acid. If suppose here is butyric acid, that is butyric. Butyric means 4 carbon. 4 carbon carboxyl is butyric. 2 carbon carboxyl is acetic. 3 carbon carboxyl is propionic. Okay. Mit, it, but, propu. You know the Latin words. It is a naphthalene group, 2 benzyl group. naphthalene. Naphthalene, it having the acetic acid. Naphthalene acetic acid, NaA. Okay, and 2-chloro, 2,4 dichlorophenoxyacetic acid. 2,4,5 trichlorophenoxyacetic acid. Okay, it all used for the cell elongation, apical dominance. Apical dominance. You know, if you cut the typical part, what will happen? Side branches will come. మనం చెట్టు గుబురుగా పెరగాలంటే ఏం చేస్తాం? టిప్ కట్ చేస్తాం. అవునా? కట్ చేస్తే ఆర్ యు దేర్?
Yes, sir.
నేనే
Yes, sir.
ఓకే. టిప్ కట్ చేస్తే బ్రాంచెస్ వచ్చే సైడ్ బ్రాంచెస్ చేస్తుంటాయి. అవునా?
Yes,
That
sir.
is the reason. Auxins at the typical part. If we cut the auxins, then what happens? Whatever the auxins are there at the tip, that will go to the branches side, bottom side. There it form the side branch. చెట్టు గుబ్బురుగా రావాలంటే మనం టిప్ కట్ చేస్తాం. ఎపికల్ డామినెన్స్ ని కట్ చేసేస్తాం. If auxins are at the tip, it goes continuously along it. Okay, and also phototropism నేను చెప్పాను కదా, లైట్ ఎటు ఉంటే అటు సైడ్ పోతుంది. జియోట్రాపిజం అండ్ ఫోటోట్రాపిజం. రూట్ ఇనిషియేషన్ కి ఇంతకుముందే చెప్పాం. ఇఫ్ యూ కట్ ఎనీ స్టెమ్ బ్రాంచ్, dip in the IAA auxin, then it form the root initiation process. Okay, and also abscission అంటే పత్రాలు ఎండి రాలిపోవటం, కొమ్మలు ఎండి రాలిపోవటం జరుగుతుంటుంది కదా, సెనెసెల్. అబ్సిషన్ ఎల్లో కలర్ గా అయి it turn yellow and then detach. That will be prevented if auxins are more. If auxins are less, more leaves will fall down, more branches will dry. Okay, and also for the parthenocarpy, that means formation of the fruit without fertilization, without the fertilization, embryology. Parthenocarpy, that means seedless fruits. Seedless fruits, we form the seedless fruit. That in the parthenocarpy also is stimulated. And also in the tissue culture also we use IAA, auxin and cytokinins we use. Auxins for the root growth, cytokinins for the stem growth. Callus, whenever we take any part from the plant as a explant, that part will form the callus, unorganized tissue. To that unorganized tissue, if we add auxins and cytokinins, it forms the root and stem. So that in the tissue culture also, to form the new plant from the tissue, any tissue, either from root to leaf, stem, branch, any part, we can produce new plant in the tissue culture. In that, we use auxin and cytokine. Okay, and also in the sex expression, ratio of the female flowers will increase if auxins are more. Okay, and already told you in the horticulture, agriculture, we use for the root cutting, and in the parthenocarpy, and also other herbicide, weedicide and herbicide. These are the 4 applications in the horticulture and agriculture. What are those? Root formation, in the tissue culture, new plants formation, and parthenocarpy, and what are they? Increasing of the flower. Okay, and weed side I already told. Next is the gibberellin. Okay, Kurosawa, a Japan scientist. You know Gibberella fujicorae. You have studied in the plant pathology.
Yes, sir.
Gibberella fujicorae. First time chadam. Buccaneer disease in the rice, paddy.
Yes, sir. Yes, sir.
Buccaneer disease. Gibberella fujicorae fungi. That Gibberella fujicorae fungi, it releases gibberellic acid. That is useful in the as a phytohormone to improve the fruit quantity and fruit size and for the bolting